Let's talk about phylogenetics. Phylogenetics is the study of evolutionary relationship between organisms. So by analyzing the DNA sequence, we can construct ancestral relationship and understand the diversification of life on Earth. So let me correct one thing. Not using the DNA sequence, we can also do this efficiently by using the protein sequences as well. So in this module, we will first of all retrieve the sequence, then blast them, and then align them, and then use all these sequence to make a phylogenetic tree. So obtaining the sequence using Biopython. Biopython, you know, it's a library that is used for the manipulation of biological data. With the help of Biopython, we can fetch sequences from online databases and analyze their properties and perform various bioinformatics tasks. So, what are the benefits? Biopython simplifies sequence retrieval and pre-processing, so making it essential tool in the field of phylogenetics. So, if we move on to our code here, which is a phylogenetic tree construction. With this code, you can construct a phylogenetic tree. You just need a list of GenBin session number, and you can find all the data that is written here in my repository. So, obtaining the sequence. We will start from a file with the accession number and country of origin. I got this accession number from here. You can see that. You can open this and then see there. So there you can choose your countries of interest. For this tutorial, I choose the sequence per continent. Thus, we will create a phylogenetic tree with 30 sequences in total. 5 sequences per continent and 5 multiplied by. So it will be 30 sequences. So what we have to do, we have to import these libraries and then we will create a FASTA file. So here you can see you can write your interest email and then open the accession number, which is your accession.txt file. If I can just show you where this file is so here you can see here are the accession number that are given here. So here is the 30 lines that are present here, and then we will open these as a CSV reader, and with the interest we will retrieve their nucleotide sequence and then print the number of sequences in file and save all these sequence in multiple FASTA file, which will be sequencegeneral.txt. So if I can show you if I got the sequences. So here you can see I got all the DNA sequence that are required to build your phylogenetic tree. So what we will do, we will move on to multiple sequential alignment. But first we will discuss that BLAST tool. What is BLAST? So BLAST is a bioinformatics tool that allows researcher to compare a query sequence against large database to identify similar sequence. So why it is important? By finding the homologous sequence, researcher can infer evolutionary relationship and functional similarities. So what role does it play, BLAST plays in phylogenetic? BLAST analysis is a fundamental step in constructing phylogenetic tree, studying the genetic conservation across different species. So how I got these sequences is by using the BLAST tool. So if we move on to BLAST tool, here we can see that. So we will set the working directory. After setting the working directory, we will obtain the BLAST result. So with this chunk of code and obtain and save the result, we will use the GenBank sequence accession number as a query. So here is my query sequence, and this is a DNA sequence we have chosen for BLAST, and which is a nucleotide BLAST. So if you like to use the protein sequence, use BLAST-P, BLAST-X, or in every BLAST possible you want to choose. So for our accession number, we have gone with the BLAST, and here is a BLAST, and then we will able to write make a code that will save the results in a file. Look at the BLAST result. So print all the results that we have, and I have set the value lower than 0.0201. You can print the sequence description, length, e-value, first piece of its alignment with the query sequence. The e-value is the number of random sequence that would have the same hit score. So as a sequence you are looking at, a low E value means the sequence found is a significant hit. So, for example, in our code, you can see we got this severe acute syndrome coronavirus, COV isolate and complete genome, which has the E value of 0.0, which is a significant hit. So it is, if it's less than that, so here you are seeing all the sequences that are present here. If it is less than that or more, if it's more than that, you can see that is not a significant hit. So that's how you can retrieve the sequence. So after retrieving, you have to al- multiple align these two, which is the pairwise alignment. So pairwise alignment is a technique of comparing two sequences to identify region of similarity and dissimilarity. What algorithm are we using? So it's a Needleman-Winshue and Smith-Waterman algorithm commonly used for pairwise sequence alignment. So by aligning the sequences, researcher can measure their evolutionary latentness and detect evolutionary changes such as their insertion, their deletions, and mutations. So moving on, you can choose to locally install or you locally install your Clustal Omega, which is a tool that we will use for multiple alignment. So if I can just show you what Clustal Omega is. Clustal Omega is a sequence alignment tool that we can use. So by using this, you can also set them to DNA and then can run this on a web server as well. And also you can install the Clustal tool as well. So okay, so if we can just see that here we have the sequence Clustal zip file which contains a Clustal file that we can use to install and run the Clustal tool as well. So we have the sequences file, we have the alignment file which is the covid.fasta, and in the Windows Python will find the Clustal Omega in the command line for you. So you just have to give this input. In Mac, Python will find this command and then Python will also find. So by using this Clustal IO will give you the result, which is the multiple sequence alignment file in your directory. It will be a FASTA file saved by the name of out/file. So after that, when we have the FASTA file, we have to draw the tree. Before drawing the actual tree, we will create a distance matrix. So this forms the basics of the tree. So we have the output file, which is the alignment.covid file, and then we will read that file. So we will read the FASTA file, and after that we will calculate the distance matrix. So what is a distance matrix you should know. When we are dealing with alignment, we have to deal with matrices as well. How much a per sequence is hitting another sequence. How much a base is hitting another base. With that value, we create a matrix like the structure. So that's what a matrix is. So there is also a matrix which is the unweighted pair group method with arithmetic mean. That is also a matrix based construction of your phylogenetic tree. So we will draw the phylogenetic tree using the upgamma, which is the unweighted pair group method with arithmetic mean method, which is used to construct your phylogenetic trees. So from our phylum, we will construct the trees, but before hitting this, we will see the, we will see that we will use this library as we have first discussed this as well. So we will import our MATLAB as well, and then we will load the tree by using the phylogen library that we first constructed. So we will set the figure size and set the size of the figure, and then we will save the image file as well. So here you can see that is the phylogenetic tree that we have constructed as well. And this is the whole tree that we have just constructed using BioPython. So if you are looking for maximum likelihood trees, you can also do that in your code. But one efficient way is to use online tools as well. Like you can use the Phylo and other phylogenetic trees like IQ tree. IQ tree is a very good tool that I have been using for a lot of time. So you can use the IQ tree to make some good phylogenetic trees. IQ tree is a tree that uses the maximum likelihood method. So if I just submit a file here, if I just submit any alignment file or fast try file, alignment, if I just open any alignment file, if I can just move, or if I just upload any file that if you just upload any file and then submit the job, let's see what is there. So obviously it will show error at this because this is a txt file, but since that I have to show you what I was doing previously. So here are the trees that are constructed by maximum likelihood. So you can just copy this consensus tree in Navic format and move to iTool. iTool is a web-based tool that is used to configure your phylogenetic trees, and then upload our tree and then just paste your Navic format and click on upload. Then you can see you can create some really good phylogenetic trees. Make it a circular tree, ignore the length, and then use a branch length, or ignore the branch length, and you can also change it to unrooted trees. You can change them to rectangular trees. You can change it to circular tree. You can add the colors. You can add length. You can add leaf ID, display node IDs. You can also type the branch or the metadata or things like that to see in your trees. So that's how you can configure a phylogenetic tree and make it look better. You can also add some specific, if for example, this is a clade, this is a group. So you can also create a color range and you can name it a group 1 file, and then you can see that you can create these all layers into different sheets, and you can say they work alone or they are highly conserved or they have some evolutionary relationship or not. So what this phylogenetic basically means. So phylogenetic helps study the speciation events, traces the origin of genetic disease, and understand the emergence of new traits. In microbiology, it aids in tracking the evolution of pathogens, studying their transmission patterns. In conservation biology, it supports effort to preserve endangered species by understanding their genetic diversity and relatedness. Phylogenetic is also valuable fields in drug discovery, biotechnology, agriculture, bioinformatics, evolutionary biology, and things like that. So what we have learned. Phylogenetic is a powerful tool to study evolutionary relationships. What insight we can gain through sequence retrieval, blast analysis, pairwise alignment, phylogenetic tree construction. Researcher can give valuable insight into evolutionary history of Earth, just like we used to do used for the COVID analysis. Understanding the phylogenetic foster breakthrough in diverse scientific discipline, contribution to advancement in biology, medicine, and beyond. So that is it for the phylogenetic tree construction by using BioPython, and by you can also do that using some really good servers that are present on the internet. And that's it for this lecture. Thank you.